Okay, so starting with this video and ending with this video, because I think it's all going to fit in one, I am using the PowerPoint that is listed in week 17. It says muscles, or chapter 11, I'm sorry, part 2. And I am all the way towards the end. I am at slide 46. So I have skipped a lot, but again, I am following the items that are listed on the schedule. And so it's taking a big chunk out of chapter 11, but I think it's going to be okay. Basically, I wanted to contrast skeletal muscle contraction with cardiac and smooth muscle. So that's what this video is going to focus on. So we're going to look at cardiac muscle briefly, and then we'll look at smooth muscle for the rest of the video. We know the differences when we look at cardiac, skeletal, and smooth under the microscope. We know that back when we studied all of our different tissue types. But let's look a little bit more at the physiology here. So we know that the cardiac muscle is involuntary. It has to contract with a regular rhythm. The cells in each chamber have to contract in unison, and the contractions have to last long enough to get the blood out of that chamber. It has to work when we're asleep, when we're awake, without fail, and without conscious attention. So these types of cells have to be highly resistant to fatigue. So the cardiac muscle cells are striated, just like skeletal muscle, but the cardiocytes are shorter and thicker. And we see those intercalated discs where the myocyte is joined to others at these notched little linkages. And on your microscope, it just looks kind of like a dark band. They are electrical gap junctions that let each myocyte directly stimulate its neighbors. So again, that's already different than what we saw in the skeletal muscle. These mechanical junctions keep the myocytes from pulling apart. That's super important as well. The sarcoplasmic reticulum is less developed. The T-tubules are larger, and they can let in extra calcium from the extracellular fluid. Damage to cardiac muscle cells repair by fibrosis. Only a little mitosis is observed following heart attacks, not really enough to regenerate the functional muscle. So there we see our cardiac muscle. Usually we can tell it apart because of the kind of extra white space in between those branches. We see the nuclei, and then there's those bands of the intercalated discs. So the cardiac muscle can contract without a nervous stimulation because it has that built-in pacemaker that sets off the wave of electrical excitation. We already talked, oh no, that's AMP2, I'm sorry. In AMP2, you will talk about the cardiac conduction of the heart, but basically the wave travels through the muscle and triggers the contraction of heart chambers. The autonomic nervous system does send nerve fibers to the heart. It can increase or decrease the heart rate and the contraction strength, but it does not initiate the muscle cell contraction. So it can kind of regulate, but doesn't initiate. The cardiac muscle sees very slow twitches. It's not quick twitches like we saw in the skeletal muscle, and it keeps the tension for 200 to 250 milliseconds, whereas skeletal muscle is much shorter than that, and that gives the heart time to get the blood out. It is using aerobic respiration almost exclusively, and that's the most efficient type because the oxygen is available. Large mitochondria for all of the ATP needed, and again, highly fatigue resistant. The cardiac muscle is very vulnerable to lack of oxygen, and again, in AMP2, you'll be able to take a closer look at that. Smooth muscle looks a little bit different to me. A lot of times when we look at smooth muscle, we don't really think it looks like muscle. We maybe think it looks like something else because we don't see those striations, but there's one smooth muscle cell right here, and then all the nuclei of all the smooth muscle cells on the picture on the right. The smooth muscle has a fusiform shape, only has one nucleus in the middle of the cell, doesn't have any striations, but the thick and thin filaments are present. They're just not lined up with each other like they are in skeletal muscle. So we don't have Z discs, but we have dense bodies. It's basically the well-ordered array of protein in the cytoplasm. The cytoplasm has a lot of cytoskeleton with intermediate filaments, and that is how the filaments are linked together, and also how they're linked to the cell membrane. No T-tubules here. The calcium comes from the extracellular fluid from calcium channels in the cell membrane. Some smooth muscles don't have a nerve supply, but others have autonomic fibers, but that's different than the somatic fibers in skeletal muscle. So let's take a look at the two types of smooth muscle. The multi-unit is first. It occurs in some of the biggest arteries and the pulmonary air passages. It's in our muscles of the hair follicle and in the iris of the eye. So taking a look at the terminal branches of the nerve fiber, they do synapse with individual myocytes. So one terminal branch to one myocyte, and each motor unit contracts independently of the others. So each terminal branch going to a separate muscle cell. And then a single unit smooth muscle is more widespread. This is what we see in most blood vessels, the digestive, respiratory, urinary, and reproductive tracts. So the myocytes here they're coupled together with each other by gap junctions. So they directly stimulate each other. And so a large number of cells contract as a single unit. Okay, this is going to get into different layers of muscle, and I really think that you're going to see more of that later in AMP2 when you get to examples. I just wanted to give you an overview. Smooth muscle is involuntary, and I wanted to be clear of how it contracts different than cardiac and skeletal muscle. It can contract without nervous stimulation. It's contracting in response to different stimuli, can be stimulated by hormones, carbon dioxide, low pH, not enough oxygen. It can also contract in response to stretch, or like being stretched, being full, if you want to think about it that way. Most smooth muscle is innervated by autonomic nerve fibers, so they can trigger and change the contraction. You can stimulate smooth muscle with acetylcholine or norepinephrine, and they can have opposite effects. I'm going to scooch past here. This just shows you a single unit smooth muscle up close. Here I just wanted to kind of look at what the contraction looks like and how it's different than a skeletal muscle. So it's still triggered by calcium. It is energized by ATP, but I'm looking at why this looks like it's two different things. I'm guessing the top one is, I don't know why it says begins a response to calcium ions that enter from the extracellular fluid through these channels. So that is different than in the skeletal muscle. Sorry, I was really thrown off by that first sentence. So the calcium here binds to thick filaments. There's no troponin, and it activates the myosin kinase, which kind of adds a phosphate to the protein on the head of the myosin, and then that hydrolyzes the ATP, or causes the ATP to break apart. The thick filaments pull on the thin ones, and the thin ones pull on the dense bodies or the membranes. So it's a little bit different than what we see in skeletal muscle. The force is transferred to the cell membrane, and the entire cell shortens. I love this analogy. It puckers and twists like someone is wringing out a wet towel. And so that's much different of a contraction than we saw in the skeletal muscle. The contraction and relaxation is very slow, so it takes much longer, and then declines over a period of 1 to 2 seconds. And then the latch bridge mechanism is resistant to fatigue. So they don't detach from the actin immediately. They don't consume any more ATP. makes most of the ATP aerobically. So here is kind of what those cells look like when they're contracted and how different they look like when they're relaxed. So I like that picture a lot. It kind of shows how different this is from skeletal muscle. And I am actually going to leave it there. We'll talk about stretching a little bit. So peristalsis, which is what you're going to see when we get to the digestive system in AMP2, it's kind of waves of contraction by food moving through the esophagus, into the stomach, small intestines, or even feces going through the colon. And so stretch can open these mechanically gated calcium channels. So basically when the muscle tissue is stretched, it can mechanically, it's not getting a signal, it's mechanically or physically opening those calcium channels to start causing the contraction. And you'll see that throughout the digestive system. Or it can be a response. Also, an example is in the bladder from the hollow organs are filling, and so when they stretch, the tissue contracts and then relaxes. It helps to prevent emptying while it's filling. That's called the stress relaxation response. So skeletal muscle really can't contract if it's overstretched, but smooth muscle can contract even when it is stretched. And then smooth muscle also has a level of plasticity. It's able to adjust the tension to the degree of stretch. Now we kind of skipped over a lot about tension, and again, that's just because I think that you're going to pick up with this in AMP2, judging by the way that Dr. Lynn had this whole thing set up. So I'm going to leave it for AMP2.